If you were to look at the data, which you wouldn't, but if you were, this is now 50 patients looking at molecular targets, expression, red is high, blue is low, and if you look at this patient 22 here, they had screaming high levels of this one drug target called FLT3, turns out to be one of the targets of sorafenib. So Deanna Mitchell et al. Treated this patient with sorafenib with acute lymphoblastic leukemia, and we saw some evidence of potential response over time. So one example of a potential responder. Another example from Al Cornelius here with clear cell sarcoma of the kidney, responding to a crazy combination of Avastin and Celebrex, which again, these are not standard of care treatments. These are off-label but tolerable, as Giselle pointed out. So a couple of examples of that. Now, the summary of all this was we believed it was feasible. The reports we provided, at least at the time, were deemed somewhat useful, but there was room for improvement, which we've done since, thanks mainly to Intervention Insights' role in this. Some responses were observed, which of course gave us encouragement and impetus to do more focused and randomized type trial designs, which is what we'll be doing with Giselle and the NMTRC. Quickly, activities we actually, you'll hear about, again, next-gen sequencing. How do we bring next-gen sequencing into this? You'll hear that from David Craig next. We're actually trying to target the tumor stem cell compartment of tumors, always refining those reports, and we have active trials both in human and, ironically, believe it or not, we're doing veterinary oncology trials in canines as well. But of course, the most focal point of today is our work, which Giselle is going to tell you about in pediatric neuroblastoma. So with that, I will finish with, the opportunity in front of us is unprecedented. The patients are still relapsed refractory patients. Unfortunately, there's an expanding drug pipeline. We have technologies that can extract data out of patients and their disease, and we have an expanding knowledge base that allows us to do a matchmaking between drugs and disease, essentially. And as Dan Von Hoff, last slide here, he's now seeing spectacular responses even in a phase 1 setting. If you treat, in this case, medulloblastoma with the appropriate targeted therapy based upon sequence analysis, in this case, whole genome sequencing of that patient's tumor, where they found a mutation in the so-called hedgehog pathway, you're seeing that type of response, which is certainly a spectacular response by anyone's book. So with that, I will pass back to Giselle, who's going to tell you about the trials.
Thank you. And thank you, Dr. Webb, for bringing us up to date on all of what goes behind the scenes. And really, I'm so thankful for Dr. Webb and his team here at the Van Andel to bring this type of analysis and technology to us to be able to help children with neuroblastoma. We started last year trying to bring this forward to our patients, and we began with a feasibility trial of molecularly guided therapy. Initially, our IRBs and et cetera asked us to really look first at 5 patients and show that we could, in real time, make therapeutic decisions based on the microarrays. And so with children with neuroblastoma, as we know, kids coming in, they're having a progression. We want to be able to do a biopsy, model, profile their tumor, and come up with therapeutic decisions. But we can't take a month or 2 to do this. We need to do this in real time to bring the therapies to the patient. So our goal was to do this, enroll, biopsy, RNA analysis, generate a report, hold a tumor board, and have it reviewed by a medical monitor in less than 2 weeks. This was our initial goal. And what was interesting about this is we have now 11 sites across the U.S. that are participating. Pediatric oncologists are coming together. Every child will have their case, their profile, their report looked at by all 11 centers. We hold an online virtual tumor board with our clinical pharmacist as well to help us create an individualized treatment plan for that patient, and that then needs to be reviewed by the medical monitor. The report, as you already saw this by Dr. Webb, builds on all these different algorithms of what is upregulated, what pathways are driving that patient's tumor, and how are they predicted. And then we're way at the bottom here in the cases. There we go. And our molecular tumor board uses this report. We have 163 drug-target interactions that we can use, and we're looking at FDA-approved drugs that we have available at our at our fingertips to be able to treat these children. The solid tumor is not only sent to the Clinical Research Laboratory, CRL, that's doing our CLIA-certified RNA expression profile, but also to the National Cancer Institute under Dr. Javed Khan to look at the next-gen sequencing for our patients. We're looking at ALK mutations in our patients to bring more options to them. And we're also analyzing bone marrow and the bone marrow neuroblastoma cells, which you'll hear about by Dr. Lisko a little bit later today. So we did these first 5 patients. I wanted to just tell you a little bit about our experience last year, which has brought us to our trial this year. These children were either relapse refractory, generally stage 3 and 4, at diagnosis. These kids have had anywhere from 2 to 13 different relapse therapies. These are multiply treated children who've been neuroblastoma generally about 5 years. And the results, we were able to, from the data biopsy through all the different steps, we're able to complete this in generally 10 to 12 days. And so this study showed us that we are able to, from the time of biopsy, analysis, report generation, tumor board, in 12 days, have an individualized treatment plan ready for our patients. This is an example, as Dr. Webb also showed, of the reports that our tumor board sees. And you can see that some drugs are predicted by multiple different mechanisms or algorithms, and those drugs that are predicted by varying algorithms as drugs that may be responsive for this patient generally get pulled up higher and looked at within our tumor board. We also get on the report the 7 different algorithms, and from each of those which drugs were predicted, which gives us quite a few options as a tumor board to create treatment plans for these patients. So these patients' tumors are also coming into our laboratory for validation. An important part of this work is going to be validating the algorithms and ensuring that they're predicting drugs accordingly. So we were able to grow all these patients' cells in culture in the laboratory. These cells are also injected into mice, and we're able to, in the initial pilot study, create mice models of each of the patients in 3 out of the 5 patients. And this can help us in further validating and testing these. So the patient tumor cells are grown in culture. We're able to look at the different drugs that were predicted and confirm that the cells are sensitive or resistant to the drugs that were predicted, and as well treat the mice with these drugs and look for responses that can help us guide our therapy for the patients. Just in also validating the RNA, we look for the quality of the RNA that's isolated from the patient's tumors, and generally the RIN has to be greater than 6.5, and we have 5 out of the 5 patients had good quality RNA. We did find ALK mutations in 2 of the patients, and this is done in the CLIA-certified lab at the Children's Hospital of Philadelphia, and so this is information we can share with the families as another option now that we have ALK inhibitor studies available. And then the next-gen sequencing that was done at the NCI by Dr. Khan. All 5 patients were able to have full next-gen sequencing done. 2 required amplification, but it was completed. The next-gen sequencing takes a little bit longer, and we're not quite using that in real time yet, but Dr. Craig will talk to us about this and how we can, in the future, incorporate this into our studies. So the outcome of our pilot study showed that the RNA was acceptable quality, the reports were generated, treatment plans were agreed and approved by a medical monitor, and everything was able to be done in less than 12 days. So we were able to move forward and work towards IRB and FDA approval in a treatment. In the first pilot study, we were not able to treat the children. And now, in our current study that we've just opened this month now, we are able to not only enroll into study, perform the analysis, and also then once we have the individualized treatment plan, we can present this to the family and move forward with therapy. We're going to be looking at safety of our treatment plans, as well as response rate and progression-free survival. As Mr. Lacey talked about, increasing our progression-free survival and good quality of life, whether it be not necessarily response, but stable disease, and improving the quality of life of these kids is really important, and one of the things that we want to be able to follow. So this is our current study design. It's just adding on to our previous, where we're actually then consenting to the families to the treatment plan and treating. We'll be doing weekly visits for safety and evaluating every 2 months for efficacy. We did have, the FDA granted us an IND as a new drug evaluation, and that was given an exemption because we were using FDA-approved drugs that have pediatric dosages. But we did require an IDE, which is an investigational device regulatory path, and that was approved by the FDA. And the investigational device, according to the FDA, is actually the gene chip, and the computer algorithms are considered a device in treating a patient. And then we obtained the Western IRB approval in January, and now have 5 IRB sites approved and open at MD Anderson Orlando, the NCI, the NIH, DeVos Children's Hospital here, Levine Children's Hospital in North Carolina, and Cardinal Glennon Children's Hospital in St. Louis. Part of what we're going to be doing in this study as well, because this is an investigational device, the FDA is requiring us to look at tumor heterogeneity and being able to replicate the report in patients' tumors. So as we isolate a patient tumor, it was now going to be, for our first 5 patients, is required to be split into 3 samples. The RNA will be isolated from the 3 samples from the same patient, and then everything will be replicated. The RNA will be isolated all the way down to 3 different tumor reports will be generated for that one patient to look at reproducibility of the device and, kind of really validating, making sure. And then also these will be frozen over time, and over time will be looked at as well. So we look at day-to-day variability analysis, and this is something that's really just going to help in terms of validate and have us move forward. The trial information is on clinicaltrials.gov, and that is the link to find out more about this clinical trial. And our contacts at the NMTRC are Genevieve Bergendale and Heidi Smith are our research coordinators and study managers, and those are the phone numbers and emails if anyone has any questions. And I can't thank enough people in terms of this is my lab from the University of Vermont and transitioning into the lab here at the Van Andel as we move forward. And Dr. Jeff Bond at the University of Vermont has been a key mentor to me in helping start the analysis, and then our whole research team at the NMTRC. And none of this study at all would ever have been possible without the support of the foundations, specifically the Owen Moscone Foundation, the Whitmer Foundation. St. Baldrick's Foundation is supporting the NMTRC infrastructure, as well as the Ishangala Foundation, Lily's Friend, and the Neuroblastoma Alliance.